2020, DeepMind shook the world with their AlphaFold tool, solving a 50-year-old problem, one that had stumped scientists for decades. To predict a protein's 3D structure just from the sequence of its amino acids, and there are at least 40 million protein sequences. It is still considered one of the greatest achievements in the history of artificial intelligence. But why does the protein structure matter so much? And how could this breakthrough change the future of medicine and the ecosystem? The human body and all complex life can be seen as a massive factory of microscopic machinery working in harmony. At the cellular level, these machines are made of proteins, each with its unique shape built to bind with other molecules and carry out specific tasks. Some perform biomechanical actions like transporting molecules within the cell or building more proteins, and others act as critical signaling molecules and structural frameworks. But how do these proteins form? The primary structure of proteins is a linear sequence of amino acids, starting with the N terminus, the amine group, and ending with the C terminus, the carboxyl group. These amino acid sequences fold spontaneously into alpha helices or beta sheets with a polypeptide backbone, and influenced by the chemical properties of the side chains and external conditions like pH, temperature, and hydration. When these secondary structures bind together, they form the tertiary structure, and tertiary structures together form the quaternary structure. Proteins could either be synthesized with a faulty amino acid sequence or folded into the wrong shape, resulting in proteinopathies like Alzheimer's and cystic fibrosis. Proteins could also enter from external agents, especially viruses. These viral proteins bind to specific human receptors to enter the cells, replicate, and disrupt normal processes. There had to be a way to study these errors. Scientists needed to uncover the rules that govern protein folding and decode the patterns behind these fatal mistakes. If we could understand that, maybe we could predict them or even prevent them. In 1973, a Nobel laureate named Christian Anfinsen proposed a revolutionary idea, one that would become the cornerstone of protein science, and it's called the Anfinsen's dogma. He believed that the protein's unique amino acid sequence was the sole blueprint needed for it to fold into its final functional shape. Through experiments, Anfinsen demonstrated that even if a protein was denatured and unfolded by heat or chemicals, it could regain its functional form when returned to the right environment. But Anfinsen didn't stop there. He revealed that the process of folding wasn't random and it was a thermodynamic one. The protein would naturally find its lowest energy state, which is the most stable conformation, and this would be its native functional shape. This idea, simple yet profound, would lay the foundation for the computational methods we rely on today. And back in time, researchers used experimental methods. X-ray crystallography, the most widely used technique, where researchers grew protein crystals and bombarded them with X-rays. The resulting diffraction pattern would reveal atomic details, allowing them to reconstruct the protein's 3D structure. Yet, it had one significant limitation. Proteins had to be in their crystalline form, which wasn't always possible. For smaller proteins, nuclear magnetic resonance could analyze proteins in their native solution state. By observing the diffusion of proteins and how the atoms interacted with magnetic fields, researchers could not only learn about the structure but also about the protein's dynamic motion. Cryo-electron microscopy, perhaps the most exciting breakthrough in recent years. This technique involves flash freezing proteins in their natural state, capturing high-resolution images of them without the need for crystallization, and then stitching those images together for a complete 3D structure. Despite knowing the amino acid sequence of countless proteins, determining its 3D structure was a daunting and labor-intensive task. And after months of work, the result would be a blurry or partial picture. And even after all these efforts, the total protein found in nature that made it to the protein data bank was just 0.1%. AI models learn to recognize patterns from evolution using multiple sequence alignments, where they observe the amino acid sequence conserved across different species. They combine this with geometric reasoning, understanding distances, angles, and orientation between the residues to infer the 3D structure. These models were trained on a massive data sets of known protein structure from databases like the Protein Data Bank, allowing them to generalize and predict new structures with accuracy. And now, DeepMind has taken their AlphaFold 2 to a more powerful AlphaFold 3, which predicts protein interactions with other molecules such as ligands, ions, and even other proteins. Give it 1, 2 or even more protein sequences in FASTA format or even other molecules, and within minutes, depending on size and compute, it gives a highly accurate tertiary structure. And with NVIDIA's BioNeMo, there's an entire AI stack of all the powerful protein sequencing models, including AlphaFold, ESMFold, GeneFarmer, all optimized to run on the GPU. This changed how we approach protein science. What once took months in a lab could now be modeled computationally in hours. With just the sequence of a target protein, researchers can model its structure, identify active sites, and simulate how a drug or compound binds to it, and design better therapeutics and even target proteins for degradation. This is more than just a breakthrough in protein science. It's a turning point in how we understand diseases, interactions, drug application, and even explore life at a molecular level. Drugs once rushed in times of crisis can be developed faster, with safety and precision, for rare and debilitating diseases that once had no answers. Personalized medicine is no longer a dream, it's becoming a reality. Therapies that are tailored to unique biology. And beyond medicine, the ripples spread even further. In agriculture and environmental science, with the understanding of protein structure, protein engineering holds the key to sustainable food, cleaner air, and smarter ecosystems.